Hi, welcome back. My name is Mrs. Horseman, and today we are going to talk about movement of molecules across the membrane. There's actually two kinds of movement across the membrane, so today we're really going to focus on passive transport, or movement that doesn't require energy. If you recall, here is a picture of the membrane, and I just want to review a couple important things that are going to help you understand how different things can move across the membrane. So remember that the membrane is two layers, and that the outside part is hydrophilic, which means it likes the water. It goes well with water. The inside portion right in here, that middle section, is hydrophobic, which means it doesn't like water, okay? So that's going to be really important when we're talking about how things are going to move across the membrane and why they need to do specific things. The membrane helps maintain homeostasis, and if you recall, homeostasis is talking about a balance between internal conditions and the external environment. And one of the ways it does that is it controls what molecules are on the inside and the outside of the membrane, and it looks at concentration. So we're going to talk a bit about that today. It is a selective barrier, so it's going to be really specific about which things can move in and out. Some things are allowed to move a lot more easily across the membrane, but some things require energy, and they have to go through specific channels or specific doors, if you will, of the membrane. The membrane also helps in cell recognition, and we're not going to spend a lot of time talking about that as a first-year biology class, but I do want to point out the glycoproteins and the glycolipids that were talked about in the last video are actually there to help identify that cell as a specific kind. So if it's a liver cell or a heart cell or a red blood cell or a white blood cell or all those other important things. Okay? All right, so let's start talking about passive transport. Passive transport means no energy is required, okay? An important key term that we're going to talk about is concentration gradient. So when you're talking about concentration gradient, we're looking at a gradual change of solutes, or parts of a solution within a given space. And I found this picture that I really liked because I think it does a good job showing what a concentration gradient looks like. So we have a high concentration of molecules, those little red dots on this side, and gradually moves to a very low concentration where there's hardly any little red dots on this side, okay? So here is high concentration, and it is a gradual or gradient change to get to low concentration, okay? So when we're talking about concentration gradient, I like this picture. So let's pretend that this is Kool-Aid, okay, that you're gonna mix in a pitcher of water. So this is the little, when you dump the little packet, it's all kind of condensed in that first little area right there. So here's the solute. All of the water molecules, these little blue things, are the solvent. So right now it's really concentrated. There's a high concentration right there of those Kool-Aid particles, okay? So as it starts to filter down through the water, it starts to move from high concentration to low concentration, okay? And then eventually we're going to get to where the concentration is equally distributed, okay? So that's all great for Kool-Aid, but when we're talking about cells, that's actually really important too. So this is showing you how that is distributed. It's going from high concentration to low concentration. Kind of makes you want to dance. All right, so that was kind of showing you in motion how particles move. In that case, it was red dye moves from a high concentration to a low concentration. It kind of evenly distributes throughout the solution. So what does it have to do with cells? Well, that's actually how particles or molecules move across the membrane. It's driven with the power of the concentration gradient so that it doesn't require the cell to use any energy. And the first way that it does that is called diffusion. And diffusion is just movement of molecules from high to low concentration. So I kind of picture it like a ball rolling down the hill. After you give the ball a little nudge, it doesn't require any energy for that ball to be rolling down the hill. It goes from high to low concentration. It's an easy movement. Here is, okay, we're using again molecules of dye, but here we've got a selectively permeable membrane, which is similar to the cell membrane, okay? So we've got a high concentration of molecules on this side, and we have a very low concentration of those dye molecules on this side. So what's going to happen is, because there's such a high concentration, those molecules don't like to be so stuck together, okay? They need their space. So what's going to happen is these molecules are going to find their way across the membrane, and they're going to start moving from high to low concentration. And they're going to continue doing that until they reach what they call equilibrium, where it's equal amount of particles on either side of that membrane, okay? So that's diffusion of one solute. And then if you look down at this picture, it's showing diffusion of two solutes. So I'm using this as an example because I like the way that there's a high concentration of the purple dye on one side and a high concentration of the orange dye on the other side. So really, anytime we're talking about concentration gradient, we're really just talking about comparison, okay? A comparison of number of molecules in one space versus the number of molecules in the other space. So as you see here, there's a high concentration on either side. In the second picture, that high concentration, the particles are starting to move across the membrane. It's not taking up any energy, okay? High to low concentration until it reaches equilibrium. So that is diffusion, the movement of molecules from high to low concentration without the use of energy. Some examples of diffusion are going to be some of the non-polar, very small particles or molecules. Oxygen and carbon dioxide are two examples of gases that move very easily across the membrane. They're non-polar, which means they work well with lipids, which are also non-polar. So they can move across that part of the membrane, which is hydrophobic, okay? All right, here is another picture, and this is actually using the cell membrane. So here we have a large or high concentration of whatever this blue molecule is. Let's say it's carbon dioxide. Okay, so here we have a large concentration of carbon dioxide, and that carbon dioxide molecules, they don't want to be right next to each other, so they're going to start to try to spread out. So they do that by moving across the membrane. Okay, they're diffusing across the membrane from high to low concentration until they reach equilibrium. All right, so that was diffusion. Now we're going to talk about osmosis. Osmosis is the diffusion of water, so we're specifically talking about water this time across the membrane. We're still moving from high to low concentration, so it still isn't using any energy, but specifically this time we're talking about water. So let's look at this picture. So we have here, inside the beaker, there's a type of selectively permeable membrane that's mimicking the cell membrane in this case, okay? So here we have, here we have a high concentration of sugar solution. So there's more sugar solution, and in comparison, there's less water solution, okay? And then on this side over here, it's more dilute sugar solution, which means there's less of a sugar concentration compared to this side. All right, so there's less sugar, more water in comparison. All right, so here we have a high concentration of water on this side of the beaker. And we're going to say that this selectively permeable membrane is only permeable to water molecules. So the sugar molecules are actually too big to move across that membrane. All right, so they're not going to be able to move across it. So what we're going to see is the movement of water molecules. So what happens is, remember, those molecules don't like to be pushed together. They like to have their space. So that water is actually going to move across the membrane. It's going to move from a high water concentration to a low water concentration. So what we're going to end up with, even though there's more water on this side, the water feels like it's evenly distributed because it's equaling out the amounts of sugar per water on each side. So what does that look like in terms of animal and plant cells? Well, there's a couple of terms that we actually have to talk about before we can look at plant and animal cells. Hypertonic, hypotonic, and isotonic. And I know those are really funny words. You've probably never heard them before. So let's take a few minutes to talk about them. So hypertonic here, it says the solution with the higher concentration of molecules. So more solute, less water. Hypertonic, less water. Okay, so when a cell is hypertonic to its environment, it has more solute on the inside. Okay, so more solute on the inside, less water on the inside. So the higher concentration of water is actually on the outside of the cell. So the water is going to move into the cell when a cell is considered to be hypertonic. Hypotonic is when the solution has a lower concentration of molecules, so there's less solute, there's actually more water. So when we were looking at that beaker that had the water and sugar solutions, the one with the less sugar solution, the least amount of sugar in that solution, was considered hypotonic. There was more water. So when a cell is hypotonic to its environment, it has less solute, more water. So that water is going to move out of the cell from high to low concentration. And I remember this because hypo to me sounds like hippo, and hippos are fat. So the cell is fat with water like a hippo. It is hypotonic. All right, the last one that I want to talk about is isotonic. Iso means same, okay? So when both solutions have the equal amounts of solute and water concentration, it's considered isotonic. So an isotonic cell, which is what your cells want to be because they like to be homeostatic, they like to be in equilibrium. An isotonic cell has an equal amount of water moving in and out of the membrane. So it has equal amounts of water inside and outside. Now I want you to understand that even though we're talking about how water moves, we're talking about net movement, okay? So here, I'm going to show you some pictures of red blood cells, okay? So here is what a normal red blood cell looks like, okay? It almost looks like a donut. We've got a little dip in the center of it. That is, red blood cells are supposed to look like that, okay? It actually allows them to better carry the oxygen to all of the different parts of our body that needs it. So in osmosis, the water is going to diffuse through that semi-selectively permeable membrane, which is the cell membrane. This red blood cell has a cell membrane just like every other cell. So in this picture, the concentration of water inside the cell is the same as the outside of the cell, okay? So inside the cell membrane, we have equal amounts of water as we do outside the cell membrane. This picture doesn't show it, but those water molecules are actually moving. They're flowing across the membrane, but they're flowing across in equal amounts. We call that net movement, okay? So even though there is water moving across in both directions, there's no net movement of water or no total movement of water. It's going to stay isotonic or equal on both sides. If you look over here, this red blood cell doesn't look very healthy. It's actually not. This is where there's a lower concentration of water outside the cell, okay? There's lower water concentration outside the cell. So right now, this cell would be called hypertonic, all right? So when that happens, when the water's leaving the cell deflates and it's going to be not very healthy, okay? In this picture, this is also not a very healthy looking cell. In this case, we have more water moving into the cell. We have a higher water concentration outside the cell, so water is going to end up moving in, and the cell's going to get really big and fat. So at this point, that cell would be considered hypotonic when there's more water inside the cell than outside, all right? We'll review those terms in class, but you need to make sure that you're familiar with them. The last one that I wanted to talk about today was facilitated diffusion. Facilitate means help, okay? So this is diffusion with help. We still have the movement of molecules from high to low concentration. We're still not using energy. The difference is this type of diffusion uses help from proteins in the membrane, okay? So a lot of times, these molecules that we're talking about that have to move across the membrane through proteins are either polar, which means they have a partial charge, okay? So they have one side that's partially positive and one side that's partially negative, so they're a little bit unbalanced in terms of electrons. When that happens, they can't cross the lipid part of the membrane, okay? Because the lipid part is nonpolar and they don't like each other very much. So they need help to get through the membrane. So they're going to use a protein, and there's two kinds. There's either a protein carrier, which it literally carries molecules across the membrane, or there's a protein channel, which is just kind of like an open door that allows molecules to freely move in and out. That's actually how water is going to move in and out of the membrane. So these molecules are either polar or they're too big to fit through that phospholipid membrane, okay? Because there's a little bit of movement between those phospholipids, but they're not far enough apart that big things can get through, like glucose, okay? So it can't move through the membrane by themselves, and they need the help of proteins to move them across that membrane. They still are moving against the gradient, so from high to low concentration, they do not use energy, okay? So here is a picture of what I'm talking about. So this is the outside of the cell out here. Here's the inside of the cell down here. And this gets a little bit more in depth than what we really need to know for this class. But what you do see is these molecules, they're insoluble. They can't get across the membrane, which means they can't get across the membrane without help because they can't get across that lipid part of the membrane. So instead, they're going to go through this channel. And this one is actually showing you a carrier protein because this comes in, it binds to that protein, the protein changes shape, so it's open here and it's going to change shape to be open here, and when it opens like that, then this molecule is going to flow through, and then that carrier protein goes back to its original shape. The other ones that I talked about were protein channels, and they're going to be open like this most of the time, okay? So those molecules can just flow right through. All right, so we talked about three pretty important things. All of those are passive transport, which means they do not use energy. We talked about diffusion, we talked about osmosis, and we talked about facilitated diffusion. And you are responsible for knowing all three of those things. And in terms of osmosis, you really need to understand hypertonic, hypotonic, and isotonic and what that means for the cell. We're going to practice those things in class, but I'm sure that there's a lot of things that you have questions about because you've never heard about some of this stuff before. So make sure you jot your questions down and we will go over them in class.